the Essex Babylab, we study how infants and young children develop and learn. Our research aims to answer questions like how do babies develop language, when do babies start to recognize themselves, or how do they learn to copy others. Studying babies is quite a big challenge because young babies can't talk. They are not very good at following instruction and have limited motor abilities. Therefore, we use indirect methods to find out what is going on inside their little heads.
What I'm studying is language comprehension, and for that we can use the eye tracker. The eye tracker works with a tiny little flashing light, an infrared light, so not visible. And what it does reflect on the baby's eye, and because of that, what the eye tracker does is measure where the eye is looking.
So another method that we use at the Essex Babylab is surface EMG. And this method makes use of the fact that whenever our muscles contract, they generate a very faint electrical signal. By placing these kinds of sensors on the baby's skin, we can pick up on this signal. And using this method, we can, for example, measure babies' tendencies to copy other people's facial expressions, even when we can't see any copying with the naked eye.
One of the methods that we use in the Babylab is EEG. Our brain cells communicate using faint electric signal, and we can measure this brain activation by placing a net like this one on the baby's head. The net has loads of little sensors that are embedded within sponges. And we soak the net in warm water with a bit of salt and baby shampoo, and this makes the sponges go soft and warm. This allows the sensors to measure the actual brain activation. Researcher will then distract the baby while the other one quickly puts the net on the baby's head. We use EEG to study a variety of different things, but we are using right now EEG to measure brain responses related to self-recognition. Near-infrared spectroscopy, or FNIRS, is a technique that allows us to find out which parts of the brain are involved in different behaviors. FNIRS uses sensors called sources and detectors, and pairs of sources and detectors are embedded within a cup like this. So it's quite comfortable for babies to wear. It's like a swimming cap, really. And the sources emit near-infrared light, which goes a little way into the head, and these lights are picked up by the detectors. And by measuring how much light is absorbed between source and detector, we can infer the color of the blood in the underlying brain areas. So if the blood is more red, then it means that there's more oxygen in the brain area, and this means that this part of the brain is more active. So we can see that the baby is engaging that part of the brain when they're watching different stimuli. And using these methods, we can study how the infant brain is processing, for example, social stimuli like faces or different sounds.
We are always looking for babies and children to take part in our studies. So if you are pregnant or have a baby or a child under 5 years old and you're interested to taking part in our research, please do sign up.